So I think poets and scientists have some things in common. The really good ones are outliers. They're, from a neurologist's point of view, being an outlier means that your brain is wired a little differently than everybody else. And I think Mary Lou and Virginia are certainly examples of outliers, and their science is really incredibly good. The other thing I've realized reading about reading is that there were writers who described things so true and so correctly from the phenomenology that ultimately it described diseases, and it described how scientific processes went about, even if that was not the specific intent of what they were doing. So I'm going to talk about Emily Dickinson's poem about moral decay, which I think really captures the scientific way that we now think about how a degenerative disease process happens. So I'm just going to say a little bit about a UCSF perspective on neurodegenerative disease, and this was really pioneered by Stan Prusiner, who is a Nobel laureate and incredible pioneer. And the idea is all these degenerative diseases that we see in age, and there are many different ones, have genetic forms and sporadic forms. And by taking the genetic forms and understanding them, scientists have been able to take the gene and place them into a worm or a cell culture and understand how that disease progresses in a very simple system. And it has three phases. The first phase is preclinical. That's before anything happens in this system. The cell is still working. Then there's an early symptomatic phase, and that happens in the case of a worm, perhaps, with a gene that causes Parkinson's, and it's not moving as quickly. And then finally, there's the symptomatic phase, and that's what we call Alzheimer's disease in humans, or frontotemporal dementia, or Parkinson's disease. And the pioneering idea that came out of UCSF from Stan Prusiner, who was an assistant professor when he came across this idea, was that all these diseases are caused by abnormal protein aggregation. One protein misfolds, and that causes a second protein that is similar in shape and structure to misfold. And these proteins aggregate, and they're in a new form that is toxic, and it kills the cell. And then I think later we've learned, and I'll come back to that they seem to spread from one cell to the next. So they don't spread randomly. This is not a random process. It is organized decay, and it is circuit specific. So this is, I'm not going to do so much science here, but I just wanted to show you a physical, visual example of what happens in Alzheimer's disease. We have one bad protein, A beta 42, doesn't belong there. It misfolds into a different shape. And then that misfolded protein grabs onto another A beta and causes it to misfold. And then you get increasingly large numbers of those bad proteins in neurons that you don't want them to be in. And I think now we think that about 11 of these misfolded proteins is what's really injurious to the cell. And then the cell attempts to compensate for this, and it starts to move them into less toxic forms and into filaments and then fibrils. And that's really what happens in Alzheimer's disease. And we can now image that end state that happens in Alzheimer's disease when these proteins have reconstituted into a slightly less toxic form. And so right now we can actually image somebody and say, yes, that bad protein is aggregated to the brain, and that's responsible for the neurodegenerative disease. It's very specific to Alzheimer's disease. It doesn't happen with any other disease. And so increasingly we're thinking about that misfolding, that process of organized decay, as a target for therapy. Of course, it doesn't happen in healthy controls either. So the story is a really long one. This is a research study done out of Washington University by Randy Bateman. And what he showed was that people who were genetically predisposed to getting Alzheimer's disease, you could start to see that slow process beginning 20 years before they were bound to get sick with the disease. So in this population, people get sick in their 40s, so he could already see the beginnings of this misfolding of the protein 20 years before there was any evidence for the problem. Around 15 years before the evidence for the problem in the person, he's starting to see on imaging that some of the neurons are not functioning quite as well with a PET scan. And then about 5 years before that person's sick, there's some evidence in the laboratory that things aren't going quite as well. Virginia and Mari-Lou study this in a really brilliant way. And then finally, the patient comes to our office with a diagnosis of Alzheimer's disease. So Bill Seeley, who, like Kay Ryan, won MacArthur Genius Award, for I think mostly for this work, has studied this process. And this, what Emily Dickinson has called an organized decay. And it's got a very interesting wrinkle that Bill figured out. And so, as I pointed out, these diseases hit different parts of the brain. So Alzheimer's disease, you can see here, tends to hit the back part of the brain, a part of the brain called the angular gyrus. On the left side, that's critical for naming. So Mari-Lou has defined a group of patients who have Alzheimer's that begins with a naming deficit. If it's the right side, it's more visual. If it's the hippocampus, it's more memory. Our patients with frontotemporal dementia who Virginia talked about have an emotional presentation. They present with problems with addiction. They don't relate well to others. You can see their caregivers find them very, very difficult. There's another group, and I'm going to say a little bit about them in just a second, that degenerate the anterior temporal lobe, and they lose semantics. People have called this semantic type of aphasia. So I think Bill showed in the first part of his study that these diseases hit very different part of the brains, and that was kind of a paradigm shift. I was taught all these diseases hit the brain together. It was just a mush. It's not true. Then the other part, I think the really brilliant part of this study was what Bill did with healthy control people. And he took something that we call the bold signal. It's a signal that is in the fMRI, evidence that that brain area is active, and asked the question in healthy young people, if I draw a region around the angular gyrus, what happens when the right angular gyrus is active? What other parts of the brain are active at the same time? He did the same thing in the frontoinsular region and asked the question, when that right frontoinsular region is active, are other parts of the brain active at the same time? And so the punchline was that each one of these diseases with a different protein hits a different circuit that is constantly active in all of us. So Alzheimer's disease hits this posterior circuit called the default mode network, which is the part of the brain that we use when we're quiet, contemplative, thinking about experiences, memories. Frontotemporal dementia hits a circuit that's activated when we're thinking about connecting to other people, doing social behaviors. The semantic dementia hits an area of the brain that is involved with understanding the meaning of words, the meaning of faces. So what Bill showed was each one of these diseases hits a different circuit that is constantly turning on and off in the brain. And so a great neuroscientist named Donald Hebb talked about the brain working in this way. The brain neurons fire together, and then they start to wire together, and then they grow together. And this has been one of the fantastic discoveries of modern neuroscience, is that, you know, if you get these neurons to fire together long enough, you can get the brain to change. And so these are the circuits that we develop as we become scientists or poets or musicians, and we are wiring up very specific circuits in the brain. Remarkably, these are the circuits that are attacked by different proteins with each of these degenerative diseases. So we are vulnerable to certain proteins that cause this specific circuit to die together. So when I was thinking about all of this, I came across this poem from Emily Dickinson. My wife writes beautiful poetry, and we read a lot together. And this is Emily Dickinson, and she wrote this, 100 years before, more than 100 years before we understood the neurodegenerative process. In fact, we've just started to understand it in this new 21st century. And here's her trying to understand moral decay, but she describes it so perfectly, so precisely, that she really captures this neurodegenerative process that goes on with these bad proteins misfolding. So crumbling, or in the case of my clinic, the patient I see with a cognitive disorder, is not an instant act, a fundamental pause. Dilapidations, processes, or organized decays. So that's exactly what Bill Seeley showed with these different degenerative diseases. They are organized decays in a specific circuit. 'Tis first a cobweb on the soul, and I think of that cobweb as a misfolded protein, a cuticle of dust, a borer in the axis, an elemental rust. Ruin is formal. The ruin in this case is the degenerative disease. Devil's work, consecutive and slow, fail in an instant, no man did, slipping, or this degenerative process is Crash's law. We see the crash, but there was this slow, steady, organized decay that went on probably for 20 years. So that's the problem and the hope of degenerative disease that I rarely think about without thinking about how Emily Dickinson described it. And she wasn't, I don't think, thinking about degenerative disease. She was thinking about moral decay. In the case of frontotemporal dementia, though, that's often what we see. It is a disease that bores right into the moral systems in our brain. So that is often the first thing that we see evidence for. So now I'm going to step into Mari-Lou's world and describe an interesting experience we've had around one of the progressive aphasias, the semantic variant. And this is Mari-Lou's nomenclature. She devised this nomenclature, and I think it's helping us develop a rational way of treating these different progressive aphasias. But these are people who, sadly, for genetic or reasons we don't understand, develop progressive degeneration in language circuits. And as Mari-Lou points out, there are different language circuits, and you can get one of these circuits depending on what bad protein you are susceptible to. If it's Alzheimer's, it hits this posterior naming circuit. If it's a type of frontotemporal dementia, with a protein called TDP43. It's this ability to devise meaning out of our world. And if it's a problem with a protein called tau, we have trouble with non-fluent speech. So this is the semantic variant, and again, this is Mari-Lou's work. It's a progressive fluent, so people talk with lots of words and lots of long sentences, but they lose the meaning of words. So this is very different than the language deficit we see in Alzheimer's disease. These patients, because it's a semantic problem, when you ask them, what is that, it's a low frequency word, they say, I don't know. But in Alzheimer's disease and most anomias, if you said it's a micro, they would go, oh yeah, microphone. These patients don't know what a microphone is. So you say, it's a microphone, and they'll go, what is a microphone? They have lost the concept of what a microphone is. So naming isn't improved by clues. They can repeat the word over and over again, but they are disconnected from the meaning of the word. If it's more on the right side, and Mari-Lou, described this as well, they lose knowledge about faces. So they lose the ability to recognize their partner, people they've worked with. And they lose, as Virginia has taught me, the ability to understand emotions in other people. So it's a very specific anatomy. It hits a very small area of the brain. It's this tiny area that is so central to poetry and writing in general. The anterior temporal lobe on the left, or sometimes more on the right. So I'm going to show you an example of a woman who is in the mild to moderate stages of the disease, and you're going to get to see her tested by Mari-Lou. So here we go. Oop.
Oh gosh.
This is a demonstration of flip phone.
She didn't know what a pencil was.
I don't know what that's called. What is it? I don't know. That yellow something or other. But I don't know. You don't know? What do you do with it? I don't play with that. Oh.
She just has a glimmer of what it is.
On the floor, and really hard, so maybe put down and bring little kids up and play with it? Is that possible? Oh.
So that's the beginning of this slow, steady loss of meaning associated with a semantic variant of the disease. There were talents, and we've described this a lot of times. Patients with this illness get tremendous visual talent. And I'll show you an example of an artist who became an artist in the setting of this disease. So just a little bit more about the phenomenology. So if you don't know what a frog is and you're asked to color it in, you're not going to color it in green. You're going to color it in maybe pink, like this gentleman did. Very typical of these patients. It's one of the ways we determine they've lost the meaning for words. The other thing that happens is the way we layer meaning onto a word, and I have a grandson, I'm watching him do this now, is lost. So animals become very prototypical. It's like they've come out of evolution. They have similar features, but they're all pretty much the same. And you can see that this person is struggling with what these words are and what they mean and how to visualize it. So the dog looks a little bit like the cat. The fish looks a lot to me like a turtle. The bird looks a little bit like a gnat to me. So I think all of these animals are, blurring together, and eventually you get one prototypic animal. This patient, I knew him in college. He became my patient. Very severe right-sided loss, and he had terrible problems with emotion, recognition of emotion, connecting with other people. And this is, I think, we realized when Victor started painting that this bothered him enormously. And he painted these couples. And I think, though, with his deficits in understanding the face, these faces show very odd combinations of emotion. And I think this captures visually the alienation that was Victor's life, alienated from understanding people and I think longing for relationships that were impossible because his emotional system was being destroyed. And then you can see the disassembly of the face as the disease spreads more posteriorly. In the last days of his life, the main pleasure he got was painting, and I think he ended up doing shapes and colors only. Very posterior parts of the brain were still accessible for this artistic process. So I thought my teacher, Frank Benson, had described the semantic variant with his teacher, Norman Geschwin, in the 1970s. But I was reading Gabriel Garcia Marquez's 100 Years of Solitude and realized he described it. And he described this disease before any scientist ever had. And so he's in this village where people get a sleeping sickness and they lose the ability to sleep, and then they develop a very, very peculiar disorder. And I'm going to read Gabriel Garcia Marquez. He discovered he had trouble remembering every object in the laboratory. He marked them, these objects, with their respective names so that all he had to do was read the inscription in order to identify them. This is the cow. She must be milked every morning so that she will produce milk, and the milk must be boiled in order to be mixed with coffee. So this is his character struggling with the meaning of what a cow is. They went on living in a reality that was slipping away, and this is just so beautiful, and it just so captures this disorder. In a reality that was slipping away, momentarily captured by words, but which would escape irremediably when they forgot the values of the written letters. And I think one way of thinking about this is Gabriel Garcia Marquez saw patients with this rare, rare disease. I don't think he did. I'll come back to that in a minute. But I think it was his struggles with writer's block, I think, that were the inspiration for this. I'll come back to that in a minute. And so this is one of our patients. And I think this, this concept of the world irremediably slipping away, captured by words, is what we see so often in our patients with this particular form of disease. And this is one of our patients that Marilu and Virginia and I all knew very well. Lovely man and lovely family. he is losing all of his words, and what he does is he's writing all of his words. He wrote all of his friends, all of his memories, and this was his sort of exercise to capture his world that was slipping away because his language was disappearing. And it reminded us so much of Gabriel Garcia Marquez's description. And then finally, just a little bit about Gabriel Garcia Marquez. Before he wrote that book, just before that piece, that gorgeous piece came out, he was living in Mexico City with writer's block and just tortured by this. He wrote essays about it. It was just extremely hard for him. And then he suddenly had a burst of verbal creativity and went to Acapulco and was able to write this book. And here's a couple of quotes I have from him throughout his life. In practice, with the experience I have, I could write a new novel without any problem, but people would realize I hadn't put my heart into it. So he was saying, in the state he was in, he could put words onto the paper, but they would be hollow, meaningless. And then he wrote around the same time, I fight head-on with each word, and almost always it is the word that ends up winning. So he wrote about this process as part of his writer's block. And then sadly, he wrote, again, I've stopped writing. He had writer's block throughout his life. So I stopped writing. 2005 has been the first year of my life when I haven't written a line. And then his brother Jamie wrote a little bit more about him. And he wrote, my brother is 85. Dementia runs in our family. He's now suffering the ravages prematurely due to the cancer that put him almost on the verge of death. Chemotherapy saved his life, but it also destroyed many neurons, many defenses and cells, and accelerated the process. But he still has the humor, joy, and enthusiasm that he always had. And so that was his story, and I think it brings up more questions than it answers. But I think what is clear to me is that Gabriel Garcia Marquez described this disease with an eloquence that we have never seen before, a decade before any scientist did. So another example of scientists seeing the world so purely, writers seeing so purely that it precedes the ability of any scientist to discover it. So writers describe the world. Their visions are unique. Sometimes they capture scientific processes, principles, centuries before scientists. We scientists have to listen to writers. There are many examples that you'll hear today. Jane's writing about Alzheimer's disease with not knowing a thing about the circuitry or the story that Virginia is starting to tell, captures exactly the phenomenology of what we see as scientists. The healing aspects of writing are important for those we care for, but scientists need to be tuned into our writing colleagues or we'll miss the universe. So that's all. Thank you.